Welcome to the deep dive. If you've ever felt like medical science is moving almost too fast to follow, well, you're definitely in the right place today. We're taking a deep dive into something really transformative in regenerative medicine. Human liver organoids. These are often called mini livers in a dish. Sounds a bit sci-fi, right?
It does, but it's very real, and these tiny structures are genuinely changing how we understand human health. The liver, I mean, it's this incredible chemical processing plant in our bodies. It does everything from making vital proteins like albumin to running the urea cycle that's key for handling waste ammonia. And maybe most importantly for our discussion, it's the main place where drugs get metabolized and detoxified.
Right, a hugely complex job. And for years, trying to study that complexity outside the body was, well, almost impossible, it seems.
Yeah.
What was the big hurdle researchers kept facing with older models?
Yeah, the limitations were pretty fundamental, both physical and biological. Yeah, people started with simple 2D cell cultures, flat layers of cells in a dish. But those just don't work well because they can't mimic the liver's real environment, the 3D structure, how cells are oriented, the different oxygen levels.
So, too simple, basically. What about using actual human liver cells, the primary hepatocytes?
Right, PHHs, primary human hepatocytes. That sounds ideal, but the problem there is availability and, well, they're incredibly short shelf life. They're hard to get, and once you have them, they lose their function really fast. Sometimes, within just 24 to 48 hours, they just sort of give up in the dish.
Okay, so they just weren't stable enough for long-term study or testing.
Exactly. Not reliable for the kind of work needed.
So that brings us to our mission for today, exploring the breakthroughs that finally allowed scientists to create stable, functional human liver models using adult tissue. And we want to understand how these mini livers are really starting to shake things up in disease modeling and drug testing.
Okay, so let's start with the core problem, which was really just about getting those adult cells to grow reliably outside the body.
Yeah, why was that so difficult? What was stopping them? Was it just about cell division?
It was tricky on a couple of levels. First, just getting adult epithelial cells, like hepatocytes, to divide more than a few times beyond what's called the Hayflick limit was a huge challenge in itself. But maybe even more critically, when researchers did manage to push them to grow, the cells often lost their way. They had this identity crisis.
An identity crisis in cells. What does that mean?
Yeah, it sounds strange, but basically the methods used to stimulate growth, especially in those older 2D systems, often activated the wrong signals, like something called YAP signaling. And this activation caused the liver cell, the hepatocyte, to undergo metaplasia.
Metaplasia. Remind us what that means in this context.
It means the cell fundamentally changes its identity. It stops being a functional liver cell and turns into something else entirely often, a kind of useless duck-like cell. So you get growth, sure, but you ended up with cells that couldn't do the liver's job anymore, a real dead end.
Ah, okay. So the growth came at the cost of function. What was this specific trick, the technical lever that finally cracked this? How did they get stable growth without the identity crisis?
The big innovation involved using a particular signaling molecule called oncostatin M, or OSM. OSM activates a pathway known as STAT3. And OSM was crucial because, unlike some other related molecules like IL-6, which only gave a temporary boost, OSM allowed for sustained long-term growth.
Okay, so OSM fuels the growth engine, but how did it solve the identity problem?
Well, that's the really clever part. Activating STAT3 specifically seemed to stabilize the cell's identity. It kept them on the right track, preventing that problematic YAP pathway and the subsequent metaplasia. So, you got the expansion you needed, but the cells remained functional hepatocytes. They kept doing liver cell things. Yeah.
That's the key difference then, expansion plus function. Okay, so we have these stable adult hepatocyte organoids, HHOs. They can grow. Now, the big question, do they actually work like a miniature liver? Can they perform those core metabolic jobs?
Yes, absolutely. The evidence shows they really do recapitulate some complex functions. For instance, they perform gluconeogenesis.
That's the liver making new sugar when you're fasting, right?
Exactly, making glucose from things other than carbs. And they also demonstrate proper glycogen storage that can store energy like a real liver, which researchers confirmed with specific staining techniques.
What about the detoxification side, waste removal?
Yep, they handle that too. They actively run the urea cycle. That's essential for converting toxic ammonia, a byproduct of protein breakdown, into less harmful urea that the body can get rid of. So that critical ammonia cleanup function, it's working in these organoids.
Okay, that's impressive metabolic capability. Let's pivot to what might be the most immediate application, drug testing. Drug-induced liver injury, or DILI, is a huge concern in pharma. How well do these HHOs predict if a drug candidate might be toxic to the liver?
They're proving very powerful for this, and the reason is they actually maintain the liver's essential drug metabolizing machinery. They express the key cytochrome P450 enzymes, the CYPs. You can think of these as the liver's workhorses for breaking down, well, something like 75% of common drugs.
Like the famous CYP3/4.
Exactly. CYP3A4 is a major one, and it's active in these HHOs, along with others like CYP2A6. Plus, they also maintain the next step, the phase 2 conjugation activities, things like sulfation and glucuronidation, which help package up drug byproducts for removal.
Right. Is there a really concrete example that shows they react realistically, like a human liver would in a real-world scenario?
Oh, definitely. The acetaminophen study is probably the most cited example. It's APP, the active ingredient in Tylenol. Overdosing on acetaminophen is a leading cause of acute liver failure. So researchers tested it on the HHOs. And here's the key part. They only needed to treat the organoids with 2 millimolar APP for 24 hours to see a clear, specific toxic response.
And why is that dose and time significant?
Because it's much closer to clinically relevant conditions. Older models often needed huge, unrealistic doses or very long exposure times to show any effect at all.
Ah, so it's more sensitive, more realistic.
Precisely. And what really sealed the deal was that they could completely block this APP toxicity in the organoids using N-acetylcysteine.
Which is the actual antidote used in hospitals for acetaminophen poisoning.
Exactly. That shows incredible fidelity. The model isn't just showing damage, it's showing the right kind of damage that responds to the right kind of treatment.
That's a very convincing piece of evidence. Okay, so if these models are going to be widely adopted, say, by drug companies, there must be some strict quality control, right? What makes a good HHO?
Absolutely. Standardization is critical for trust and reproducibility. There are defined quantitative benchmarks. For instance, you need to prove they're making the right proteins. They have to consistently secrete key liver proteins, like albumin, ALB, and alpha-1 antitrypsin, AAT. Above a certain level, the benchmark is often set at over 1 microgram per 24 hours per million cells.
Okay, so that's checking their protein output. What about checking if their drug processing factories, the CYPs, are actually working properly?
Right, you need to check their function. The CYP enzyme activity has to be inducible. For example, if you expose the organoids to a known inducer, like rifampicin, you need to see a significant jump, say, more than a 1.5-fold increase in CYP3A4 activity.
So you can switch the machinery on, essentially.
Yes, it shows the regulatory pathways are intact. And beyond these numbers, there are qualitative checks, too. Researchers look for the formation of tiny channels called biocanaliculæ using specific dyes, like CDFDA staining. And they also check the organoid's ability to clear substances, often using a dye called indocyanine green, or ICG, watching how quickly it's taken up and then released.
So a whole battery of tests to ensure they're not just growing, but really functioning like mini livers.
Correct. Rigor is absolutely essential.
Okay, we've seen how well they model basic functions and drug toxicity. Let's look at how they're being used to tackle specific complex liver diseases. Maybe start with genetic disorders?
Yeah, the monogenic diseases caused by a single gene defect are a great application. Alpha-1 antitrypsin deficiency, AATD, is a prime example. Here, researchers can take a tiny biopsy from a patient with ATD or even use stem cells reprogrammed from their skin and grow liver organoids. And these patient-derived organoids accurately mimic the disease hallmark. The AT protein gets made, but it misfolds and gets stuck inside the liver cells, causing damage instead of being secreted properly.
Wow, so you have a personalized disease model in a dish. Can you then use it to test potential therapies for that specific patient?
Absolutely, that's one of the most exciting aspects. These ATD organoids have been used to screen libraries of compounds, looking for drugs that might help the protein fold correctly or clear out the clumps.
Yeah.
And even more advanced, researchers have used gene editing tools, like CRISPR-Cas9, directly on these patient organoids to correct the underlying genetic defect and actually rescue the function at the cellular level.
That's incredible. Gene therapy tested directly on a model of the patient's own tissue. Okay, let's shift gears to metabolic diseases, which are hugely prevalent. Things like fatty liver disease, NAFLD, and its more severe form, NASH. These are complex, often driven by lifestyle factors. Modeling that must require more than just the main liver cells, right?
You're absolutely right. To really capture the complexity of a NASH progression, which involves fat accumulation, inflammation, and scarring or fibrosis, you need more complex models. So researchers are now using multicellular organoids. These include not just the hepatocytes, but also other crucial liver cell types, like stellate cells, which are the main drivers of fibrosis.
The scar tissue cells.
Exactly. And also Kupffer-like cells, which represent the liver's resident immune cells. When you expose these more complex multicellular organoids to triggers like excess free fatty acids, mimicking a high-fat diet, they actually mirror the key stages of NET's progression. You see the fat buildup, steatosis, signs of inflammation, and crucially, the beginnings of fibrosis.
That's a major step towards modeling lifestyle-driven diseases. Did they notice any specific structural changes in the organoids that matched what happens in human patients?
Yes, and this is a really interesting finding. In the NH model organoids, they observed subtle but specific changes to the network of bile canaliculi, those tiny bile drainage channels we mentioned earlier. They became narrower and shorter. And this structural change is actually known to occur in the livers of human patients in the early stages of NETH. So the model is picking up on these early pathological signatures.
Really detailed mimicry. What about infectious diseases? The source material mentioned organoids are much better for virus studies than the old duty cultures.
Yeah, they provide a much more realistic home for viruses. Hepatitis B virus, HBV, is a great example. Infecting traditional cell cultures with HBV was often very inefficient, but infection works much better in the 3D organoid environment. And, importantly, the organoids support the entire HBV life cycle, including the formation of something called cccDNA, that's the persistent viral template hiding in the nucleus that makes HBV so hard to cure.
So you can study the whole process, including the tricky bits.
Right. And organoids derived from HBV patients or infected in the lab show predictable dose-dependent responses to antiviral drugs. This supports their use for testing new therapies or even personalizing treatment based on how an individual's mini liver responds. Researchers even used them during the COVID pandemic. They found that organoids derived from NASH patients were actually more susceptible to SARS-CoV-2 infection, likely because of the underlying chronic inflammation.
Fascinating link there. Okay, so let's try and synthesize this. If these organoids can be derived from individual patients and maintain their genetic and functional characteristics, they really are like personalized patient avatars, aren't they?
That's exactly the term people are using, and it captures the potential perfectly. It's about creating a test bed that reflects your specific biology.
It feels like we're right on the cusp of truly personalized medicine here.
We are making huge strides, but, it's important to keep the current limitations in mind as well. These organoids, while amazing, aren't perfect replicas of a full liver just yet.
Okay, so what are the main missing pieces right now? What can't they model effectively yet?
Well, two of the biggest missing elements are, first, a proper functioning vascular system, blood vessels integrated into the structure. And second, the full complexity of the immune system, particularly circulating immune cells that would normally travel to the liver.
Right, no blood flow and limited immune interaction. Why are those so critical? What kind of liver problems depend heavily on those missing parts?
They're crucial because their absence makes it very difficult to model diseases where the immune system plays a central complex role interacting with the liver. A key example is a type of unpredictable drug reaction called idiosyncratic DILI, or IDLI. We often don't know why only certain people react badly to a specific drug, and it frequently involves a complex interplay between the drug, the liver cells, and the patient's unique immune system.
So modeling that kind of rare immune-driven reaction is still out of reach for current organoid tech.
Largely, yes. Integrating realistic blood flow and a dynamic immune component, that's really the next major frontier in organoid bioengineering.
That makes sense. It sets the stage nicely for what needs to happen next. Which brings us to our final thought for today. Looking ahead, once some of these hurdles like vascularization and immune integration are overcome, what's the ultimate vision? Where could this technology take us?
I think the really transformative potential lies in scale and data. Imagine large-scale organoid biobanks. We're talking about potentially thousands of organoid lines derived from a diverse population of patients with different liver diseases, different genetic backgrounds.
The library of personalized mini-livers.
Exactly. Having that resource would revolutionize pharmacogenetics, the study of how genes affect a person's response to drugs. Researchers could finally draw robust correlations between specific genetic variations, disease progression in the organoid, and, crucially, how that patient's avatar responds to different drugs, including those idiosyncratic reactions.
So you could genuinely predict, based on someone's genetics modeled in their organoid, whether they're likely to have a bad reaction before they even take the medication.
That's the ultimate goal.
Sure.
Moving from population averages to truly individualized prediction and treatment. It sounds futuristic, but the foundation is being laid right now with these organoid models. It's about making precision medicine a widespread reality.